Welcome back, everyone. Today, we're getting right down to the absolute foundation of how your cells operate. We're talking about the master messenger of the microscopic world. Okay, let's dive into this and jump straight into a real-world biology problem, the exact kind of puzzle you're likely going to encounter on your path to a healthcare career. So we're tackling an actual ATI TEAS version 7 science question today. It's question 39 of 50, and it asks, which of the following molecules acts as a messenger that carries genetic information from the nucleus to ribosomes? Now, this is a classic, really foundational question about eukaryotic cells. You know, those complex cells with a defined nucleus, literally the exact same kind of cells that make up your own body. And we aren't just going to memorize an answer key here. We're going to break down exactly how to approach this so you completely understand the beautiful biological mechanism behind it. Looking at our 4 possible multiple-choice options from the source material, we've got lipids, DNA, RNA, and proteins. But hey, before we rush to an answer, let's take a little micro-journey and logically eliminate the incorrect ones. First off, we can immediately cross off lipids, right? Those are basically just fats used for energy storage and building cell membranes. And proteins? Well, they're the actual structural end products. They're the heavy lifting machines doing all the work in the cell, not the messengers carrying the original genetic code. So process of elimination leaves us with the 2 famous nucleic acids, DNA and RNA. So which one is the true messenger? Let's look at DNA first. What's really fascinating here is that you can imagine the nucleus of a cell as a highly secure, locked vault. Your DNA contains all the vital genetic information necessary to build and maintain you. It's literally the master blueprint. But because it's so incredibly precious and massive, honestly, it is permanently locked away inside this nuclear vault. It just cannot leave. So think about it. If the DNA is trapped inside the nucleus, but the protein-building factories, the ribosomes, are located outside in the cytoplasm, we've got a major logistical problem. The cell desperately needs a designated runner to carry those instructions outward. And that brings us to transcription. This is the first crucial biological step to solve our cell's communication problem. Transcription is where that locked-away DNA code is accessed and successfully copied into a brand new, highly mobile molecule called RNA. It's kind of like taking a photograph of a rare ancient reference book in a library because, well, you aren't allowed to check the actual book out. The RNA is that photograph. It's a single-stranded, lightweight copy of the exact specific gene that the cell needs right at that very moment. Let's move to and see how this builds, because now we can actually trace this newly created messenger's incredible journey. Step 1, the DNA sits securely in the nucleus. Step 2, as we just saw, transcription creates our RNA copy. Then comes step 3, the physical travel. Because it's a lightweight, single-stranded molecule, this RNA can actually physically travel right out of the nucleus. It literally slips through these tiny security doors called nuclear pores. It navigates through the cellular fluid, seeking out its final destination. And finally, step 4 brings it right to the doorstep of the ribosomes for the next phase. Translation. So, the absolutely crucial point here is what happens upon arrival. Once that RNA hits the cellular assembly line, the ribosomes, we enter the phase called translation. The ribosomes literally clamp right onto the RNA molecule and read it. They use this carried genetic code to string together amino acids, efficiently converting that message into functional working proteins. And you know what's truly mind-blowing? The speed. A single ribosome can add amino acids at a staggering rate, sometimes building an entire protein chain in just a few seconds. It is an incredibly fast, highly efficient, microscopic factory. Without this final translation step at the ribosome, the DNA's brilliant instructions would just sit silently and uselessly inside that nuclear vault forever. Coming back to our ATI TEAS test question, everything suddenly clicks into place, and this brilliantly illustrates why RNA is the undisputed correct answer. Think back. We logically eliminated lipids and proteins right off the bat. We established that DNA is permanently trapped in the nucleus. Therefore, RNA is the only molecule that literally acts as that necessary messenger. It completely bridges the physical gap between the tightly guarded DNA in the nucleus and the protein manufacturing ribosomes out in the cell cytoplasm. If you're tackling exams like the TEAS, mastering these foundational concepts is absolutely critical to your success. Our source material for today's question actually originates from cheatproctoredexams.com, which is a service offering personalized tutoring for proctored exams. But hey, beyond just passing a test, I want to leave you with this. Considering that trillions of cells in your body are successfully executing this exact RNA messenger process right at this very second, how might fully understanding these tiny, microscopic journeys be the ultimate key to unlocking your entire healthcare career? Think about it.
Keep learning, keep focusing on the fundamentals, and I'll catch you in the next